There's a creature in the ocean right now that has more in common with a genetic engineer than with any animal we'd consider its close relative. It's not a fish. It's not a mammal. It's an octopus, and the thing it can do to its own nervous system is so strange that when scientists first mapped it out, they had to double-check their data. Octopuses, squid, and cuttlefish rewrite roughly 60% of the RNA in their brains before that RNA ever builds a single protein. Humans, we edit less than 1%. And what makes it even weirder is that they crank up the editing when the water gets cold, like they're literally tuning their nervous system to match the temperature outside. You and I split from these animals about 500 million years ago. Somewhere along the way, our branch of the tree forgot how to do this. Or maybe never figured it out in the first place. About 500 million years before any of this was understood, a small, soft-bodied animal in a warm prehistoric sea wandered off in a direction that would eventually produce octopuses. Another wandered the other way and eventually produced you. Both branches have nervous systems. Both branches build proteins out of DNA. The difference is what they do between those two steps. And the cephalopod branch has been quietly running an experiment on that middle step for 500 million years. To understand what they're doing, you first have to remember what most animals do with their genes. DNA is the master file. RNA is the working copy. When a cell needs to build a protein, an enzyme reads the relevant stretch of DNA and produces an RNA transcript, which is basically a sentence written in the same alphabet. That RNA sentence then goes to the protein factories in the cell, and the factory reads it and assembles the protein letter by letter. The whole point of the system is that the message is supposed to be a faithful copy. If you change the DNA, you can break the animal. If you keep the DNA stable and never edit the message, you get a reliable, slow-moving species. Cephalopods do not respect that rule. Inside the neurons of an octopus, an enzyme called ADAR walks along the freshly written RNA and changes specific letters before the protein gets built. The change is small chemically. It swaps a single base called adenosine for a related one called inosine, and the protein-building machinery reads that inosine as if it were a different letter entirely. The DNA itself stays untouched. The change happens only on the message in transit. Run that swap thousands of times across thousands of transcripts, and you have a nervous system that is, quietly, not quite the one the DNA is describing. The scale of this in cephalopods broke previous assumptions about what RNA editing was for. According to a review in Briefings in Functional Genomics, vertebrates like us use this same A to I editing too, but it touches less than 1% of our brain transcripts and tends to hit non-coding regions where it doesn't change a protein. In cephalopods, the editing lands on protein-coding regions, in their brains, in their nervous systems, at a rate that swallows most of the transcriptome. They are not nudging their biology. They are rewriting it on the fly. The trigger for the rewrites turned out to be water temperature. A team at the Marine Biological Laboratory in Woods Hole, led by Joshua Rosenthal, ran an experiment that sounds almost too clean. They took octopuses and dropped the water temperature on some of them, while keeping others in warmer tanks, and then they sequenced what the animal's neurons were doing. According to their 2023 paper published in Cell, the cold water octopuses showed 13,285 sites of active RNA editing in their neural tissue. The warm water octopuses showed 550. Same animal, same DNA. Two different nervous systems sculpted by water temperature. That gap is not subtle. That is the difference between a small tweak and a wholesale renovation. The cephalopod brain in cold water is editing itself at a level our brains have never approached. The proteins this affects are not random. The cell paper showed editing hitting genes that build the actual machinery of neurons, including a protein called Synaptotagmin, which is part of how neurons release the chemical signals they fire at each other, and another called Kinesin 1, which is a kind of molecular freight truck that hauls cargo along the inside of long nerve cells. Editing those proteins doesn't just change the animal's body in the abstract. It changes the speed and the timing of the signals running through its nervous system. A cold-tuned octopus is, at the protein level, running on slightly different wetware than a warm one. The strangeness of this adaptation is that it doesn't need generations. A normal animal adapts to cold over many thousands of years by surviving and breeding and slowly shifting its DNA. A cephalopod adapts in the hours it takes for ADAR to walk along enough transcripts. When the temperature drops, the editing rate climbs and the neurons retune. The animal is, in some real way, a slightly different animal by the next morning. If you're enjoying this little guy as much as I am, hit subscribe. More stories like this on the channel every week. There is a trade-off attached to all of this, and it might explain something biologists have been arguing about for years. If a species can fix its biological problems by editing the message instead of the master file gets very little reason to change. The DNA stops feeling the usual evolutionary pressure to adapt, because the RNA layer is doing the adapting for it. Research summarized by the University of Chicago has floated the idea that this is exactly what has happened in cephalopods. Their genomes evolve more slowly in certain regions than you might expect, especially around the genes that get edited the most. The RNA system has, in effect, taken over the day job that DNA mutation handles in most animals. That hypothesis hasn't been settled, but it lines up with something biologists have noticed about cephalopods for a long time. They are weirdly stable as a group across deep time. The body plan of a modern octopus would be recognizable to someone holding a Cretaceous fossil. Their nervous systems are extraordinarily complex, but the underlying lineage is conservative. A creature that can retune its neurons in an afternoon doesn't necessarily need to mutate its way into a new species every few million years. It also offers a possible angle on the famous cephalopod intelligence, the lockpicking, the tool use, the problem-solving that has made octopuses minor internet celebrities for a decade now. Their brains are already strange in structure. Most of an octopus's neurons are not in its central brain at all, but spread out through its arms. Each arm runs a lot of its own processing locally. Adding heavy RNA editing on top of that means each of those distributed nerve clusters can in principle tune itself in real time to whatever the arm is currently doing. Crawling into a cold crevice, squeezing through a jar, tasting a new chemical signal in the water. The wetware is not a fixed object. It is a moving target. And from the south side, we are catching up to it. Science News reported on a follow-up to the Rosenthal lab work that has pulled in researchers across a dozen other institutions, all trying to map exactly which transcripts get edited under which conditions and what the resulting proteins do. A picture is starting to form of cephalopods as animals that have basically invented a second layer of inheritance, one that doesn't pass through eggs and sperm and generations, but through enzymes acting on messages inside individual cells inside one lifetime. There is something philosophically odd about all of this. We tend to think of an animal as a fixed thing built from a fixed instruction set. The DNA is the recipe, the body is the dish, end of story. Cephalopods quietly demolish that picture. The recipe goes in. Then a small army of editing enzymes leans over the page and crosses out words and writes in new ones based on the kitchen temperature. The dish that lands on the table is related to the recipe, but it is not exactly the recipe. And tomorrow's dish from the same recipe in a colder kitchen will be different again. Our branch of the tree never developed that habit. Maybe the early vertebrates ran a version of the experiment and found it wasn't worth the chemical cost. Maybe they leaned harder on body temperature regulation instead, especially the mammals and birds that learned to keep themselves warm from the inside, which would have made RNA editing as a thermal tuning trick mostly pointless. Maybe the cephalopod path is one of those evolutionary roads that only stayed open for a single lineage out of all the soft-bodied animals that ever existed. And we are looking at the descendants of the only survivors who really committed to it. What we know for certain is the result. A creature in cold water tonight, somewhere off a rocky coast, is sitting in its den with a nervous system that is genuinely not the one its DNA describes. Its neurons are running a custom build, produced by enzymes that have been chewing through its own RNA for the past several hours. By the time the sun comes up and the water warms a few degrees, the build will start sliding back the other way. The animal is, in the truest sense, a self-editing machine. And it has been doing this for the entire stretch of time that vertebrates have been climbing out of the sea and rearranging their lives on land. The honest takeaway isn't that octopuses are smarter than us, or weirder or older, although they are arguably all three. It's that there is more than one way to be an animal. The path our ancestors took 500 million years ago is one solution to the problem of staying alive in a changing world. The path the cephalopods took is another. Both work. Both produce nervous systems. One of them, every cold night in the ocean, is rewriting itself. If you found this fun, there are more stories like this on the channel. See you in the next video.